Okay, I'd like to welcome everyone back to the second day of our meeting, applying 20th century toxicology to green chemistry and material design. And the thrust of this, again, is really looking upstream in terms of product design. I'm used to thinking of downstream from a regulatory perspective. So it really is a shift in thinking for some of us. And we're going to build on yesterday's discussion. Paul Anastas opened the meeting by reminding us that hazard is a design flaw, that we need to turn to the intrinsic properties of chemistry to think about better design of chemicals up front. And in his view, we were really facing a problem of scale up, because there are many examples, good examples, of green by design in the literature. And we heard from Helen Holder in terms of the design chain, choosing chemicals with less intrinsic hazard properties. We heard from Helen about a framework for doing that. And we also heard from Mark Thompson of DuPont about designing chemistries very upfront. And he gave us some very good examples, one in particular of a agricultural chemical. And then even in the pharmaceutical industry, Russell Navin shared with us some of their approach to formalized ways of looking at creating chemicals with less hazardous properties. And we had a series of very informative talks, presentations, looking at new tools from a variety of levels of biological observation with a variety of depth, the interaction between biology and the molecule. And we also heard about ways of just looking at chemical properties, and from those chemical properties sorting through what is less and more hazardous. Adelina's talk, we also heard from someone in the audience, Ruben, who gave us a series of presentations looking at docking with receptors. So after that, we had a very rich discussion about bridging. Lots of analogies around building that bridge between the newer toxicology and the older, and how do we move along that somewhat shaky bridge at the moment. So lots of discussion around matching the tools that we have to the problem at hand. Again, thinking upstream. And today we're going to pick up on those issues by looking at how we're building some of the emerging toxicology tools into product design. What kinds of frameworks can we adopt to advance this? How can this be done throughout the life cycle of the product, including from considerations of the product itself to solvents and catalysts used in product manufacture? How do we move more towards this definition that Anastas started the day with for green chemistry, which is producing and using less hazardous chemicals and moving towards safer chemicals? So what kind of criteria might we consider for devising more greener products? How can this be done in a way so that we can evolve the way in which we approach the problem as we gain more knowledge with the tools and as we develop these tools further? And how can we begin to balance the toxicology considerations like energy use, carbon footprint, and so forth with these toxicological considerations? So that's that's the morning in front of us. And we have a very great lineup of speakers with expertise in toxicology. So we're going to start off our discussion with a presentation from Calbert Anderson. It's a pleasure to have her with us. She's been advancing green chemistry tools at the US EPA. She comes at it with an experience in the NGO community and the consulting world. So we're going to hear from Cal about some frameworks and begin to think the extent to which we can use such frameworks, potentially build on what we heard from yesterday, or do we need something new? So Cal, welcome.
I think I'll stick with the podium, if that works. And Lauren, you'll be timing me, so you'll let me know if I start to run late? Okay, great, thanks. Well, thank you so much for the opportunity to be here today to talk about my work in design for the environment. I was asked to discuss criteria frameworks that we use to evaluate chemicals for safety, safety being based on the hazard properties of the chemicals, and whether these frameworks are applicable to data that we'll be seeing from new modes of toxicity testing. So I need to start, of course, with the disclaimer. The views expressed in this presentation are mine and not necessarily those of my employer, the US EPA. Now, yesterday, of course, we heard from Paul Anastas and about the importance of green chemistry at EPA. And within my home office, which is the Office of Pollution Prevention and Toxics within the Office of Chemical Safety and Pollution Prevention, we have several different programs that address green chemistry, such as the Presidential Green Chemistry Challenge Awards. We have a green engineering program that's growing in scope. And we have the Design for the Environment program, or DFE, which is what I work in. Just as background, Design for the Environment is interesting. It's a little different. It consists of voluntary programs as opposed to regulatory programs. And the purpose is to provide incentives for the development of safer products. And when we talk about product safety, we really are focused on the chemicals that are used in those products and whether they are safer, i.e. Low hazard, and we look for incentives to build this into product design. We also have life cycle analysis capability, as well as a best practices program for when safer alternatives are not available. The tools that we use and develop are rooted in the expertise within EPA regulatory programs and the 30 years of evaluating chemicals in commerce. And a lot of expertise has been developed over these years. And so we rely heavily on this expertise in our evaluation of chemicals. But because we're a voluntary program, we also rely on multi-stakeholder partnerships, and so that is an important aspect of all of the programs that we have. So, for example, the 4 major components of Design for the Environment include the Safer Product Labeling Program, where we allow the use of the Design for the Environment logo on products that are made with safer low-hazard chemicals. We have a chemical alternatives assessment program, and the goal of that is to identify and evaluate alternatives to EPA priority chemicals, and we base that evaluation on a range of human health and environmental attributes. And the goal is to promote informed substitution, so we avoid switching to a chemical that has perhaps no advantage from a hazard standpoint. As I mentioned, we do have a life cycle assessment program. Life cycle assessment, LCA, is an important tool, and in particular for when there are concerns for a product or a material throughout the product life cycle. And best practices, of course, when there are no options, it's really important to control exposures to workers, consumers, in the environment, and developing approaches to do that is part of our capacity. Now, yesterday we saw several times the 12 principles of green chemistry, and I just want to emphasize that our focus on hazard and the intrinsic properties, hazard properties of chemicals is really rooted in these 12 principles, 4 of which directly address the need to move to lower hazard alternatives. Another context, I think, that's really important to understand how Design for the Environment operates is the understanding that people are making decisions about what chemicals they use every single day. And we don't always have all the data we want to know about a chemical in order to evaluate. But to paraphrase a notorious quote, we evaluate the chemicals with the data that we have, not necessarily the data that we wish we had. We have to do it, and we have to help inform decisions. So DFE, our goal is to make available the information, the best information that is available for the chemicals that we're considering in a comparative framework. So that we actually help our stakeholders, because remember, we're voluntary programs, we work in partnership. So we're helping our stakeholders make the best decisions they can given the information that's available. And I should point out, it's down at the bottom, but it's really important. Of course, the alternatives that we consider have to be functional. There's no point in identifying a safer alternative if it's not going to work. So functionality is a critical aspect of what we do. Now, so we talk about safer. We want safer chemicals. We want low hazard chemicals. Hazard, of course, can be ranked on a continuum. And I think this continuum is very much a part of what we do. Now, obviously, benign by design, low hazard chemicals will be found on the safer end of the continuum. The high hazard chemicals, the ones we're looking to substitute, will be found on the left side of the continuum. Often, we're not dealing with like truly green design chemicals, but we're operating in this middle zone where we're looking for incremental improvements. Now, the criteria used to define safer can be used de facto as toxicological criteria as part of the green chemistry criteria. Now, this concept of hazard on a continuum is not new. The EPA, Office of Pollution Prevention and Toxics, has been evaluating and kind of ranking chemicals for many, many years, both within the new chemicals program, existing chemicals program. Certainly DFE is using this. The Office of Pesticide Programs ranks hazard, has criteria for ranking hazard. European Union REACH Annex 4 identifies criteria that for substances of very low concern, so that's another way of evaluating chemicals. And the globally harmonized system of classification and labeling, that's another set of criteria that can be used to evaluate and assess hazard. And so next I'll turn to some examples of how criteria are structured so we can see what the different options may be. So yeah, there. So first, the globally harmonized system. There are two basic ways of classifying or developing criteria to assess hazard. The first is threshold-based, using actual dose cutoff values, and the second is evidence-based. And the evidence-based relies on the strength of the evidence in order to classify a chemical as a known, presumed or suspected hazard, and those are terms that are often used in this type of criteria. And often this is used for endpoints such as cancer, mutagenicity. GHS also uses it for reproductive toxicity. But the threshold-based criteria rely on actual cut points that are based, a couple things, they're based on animal doses or animal exposure concentrations. So they're very much married to animal testing. So in theory, you would take a low AL or no AL and by route of exposure, and you would compare to the threshold criteria, and you would be able to assess whether it's a high, moderate, low concern for that particular endpoint. And this example is based, these are the repeated dose guidance values that GHS uses, and it would be like a 90-day animal study that you apply to this criteria to. So EPA uses both types of criteria, the evidence-based and the threshold-based, and so you can use like assessing hazard using this strength of the evidence. in one way to apply it, you need quite a bit of data. So you're looking at kind of the strength of the evidence for a carcinogen. Is it a known human carcinogen? You need quite a bit of data to assess that. But because it's like a relative scale, you can also apply it to chemicals with very little data. There's a structural alert that might raise the concern to moderate over rather than low. So it's kind of a flexible scale that may have some utility. Now, the threshold-based criteria, you need data because you can't evaluate against a threshold unless you actually have data. So it requires that you have some type of animal data in order to compare to these threshold cut points. And so DFE, we use threshold-based criteria for these endpoints. What I have shown here are the reproductive and developmental toxicity criteria that we apply in our alternatives assessment framework, just as an example. And once again, it's based on animal doses or animal exposures, and it's by route of exposure. And these cut points, obviously, we would apply a low AL or no AL, and the higher the dose, you need to see some type of adverse effect. The lower concern you might have for that endpoint. And just as an example, there may be a chemical that is a known human developmental toxicant, so that might rank kind of a high concern from an evidence-based framework. But if the no AL is 750 milligrams per kilograms, that would rank as a low concern based on a threshold based framework. So you can see that there are the different frameworks you apply matter. They'll give you different results. And there are, obviously you can make combinations of use both criteria. There are different ways that you can construct your criteria. I think what DFE adds to this is, rather than looking at the universe of chemicals and ranking them and evaluating them, we divide the chemicals by chemical function. So we're comparing, because basically we're asking the question, what are the alternatives and how do they compare? So we're comparing solvents to solvents and surfactants to surfactants. We're not comparing a plasticizer to a pesticide. So we are narrowing our scope and asking very focused questions, which chemical is preferable from a hazard standpoint. And one other point that to make is that the criteria can be tailored to the functional class to distinguish safer chemicals. So the criteria you might design for a solvent may be somewhat different than what you would design to compare surfactants. And so that's the idea behind this. Now, so for example, we'd love to know everything about a chemical and the chemicals that we're evaluating. But the bottom line is some endpoints are not particularly distinguishing. They don't help us actually determine which is a preferable chemical. And as an example, surfactants in general are toxic to aquatic organisms. And so if you compare surfactants based on aquatic toxicity, you're not going to see very many differences. However, some surfactants degrade extremely rapidly and others do not. And so the preferable surfactant may exhibit some aquatic toxicity, but it also will degrade rapidly to mitigate some of that concern. There may be solvents that in repeated dose toxicity testing exhibit very low levels of hepatotoxicity, and you can't really tell them apart. So you would look to other endpoints like developmental toxicity in order to distinguish a preferable solvent along that hazard continuum. Once again, we may not be talking about benign by design, but we're working in that kind of middle ground to kind of move in the direction towards inherently safer. point out that we do consider life cycle even when we are evaluating chemicals based on hazard. And one of the points I want to emphasize is something that Paul Anastas mentioned yesterday, and that there are benefits to switching to low hazard alternatives that resonate throughout the product life cycle, and that is reduced concern for worker exposures or accidental releases, reduction in the need for engineering control, storage, handling, and transport costs, as well as reduction in concern for potential consumer exposures and disposal. So next I want to give two examples from two of our programs, Safer Product Labeling Program and Alternatives Assessment Program. We use our criteria a little differently in each program, and so once again it illustrates different approaches to developing and applying criteria. And then I'll turn to kind of the meat of the talk, which is what we do with this new data that we're getting. So the Safer Product Labeling Program, the goal is to provide an incentive for formulators and manufacturers to move away from hazardous chemicals towards inherently safer alternatives. We work closely with stakeholders in order to identify substitutes that meet our criteria. And our criteria have, in fact, become kind of design criteria for in some companies. One of the distinctions about our approach in the Safer Product Labeling Program, because we're labeling products and we're saying these products are safer, our criteria are actually pass-fail criteria. So, for example, these are 3 endpoints just to illustrate what we might use for carcinogenicity. Basically, this is one of those weight of evidence or evidence-based criterion. We do not allow known, presumed, or suspected human carcinogens in products that are labeled through the Safer Product Labeling Program. But with our developmental toxicity and repeated dose toxicity endpoints, these are threshold-based. So if chemicals with animal testing or an analog, a suitable analog, actually demonstrates that based on testing, the chemical passes our criteria, then it would be allowed in a labeled product. Now, this is very different from our alternatives assessment approach, which is really more of a compare and contrast program. The goal of this is to identify and evaluate potentially safer alternatives and make this information available to our stakeholders so that they can make decisions about what alternative might be a good fit for them. And so the outcome is this compare and contrast format where we provide information on a broad range of human health and environmental endpoints. So, and Helen showed a snippet of a table like this yesterday. This is a table that compares flame retardants. A couple things to note. We evaluate a range, and this is a summary table. We do provide actually the details of how we arrive at these hazard calls in the documentation that we provide with this. But in summary, we evaluate a range of human health and environmental impacts. The cancer, for example, would be more evidence-based. It's based on the strength of the evidence for carcinogenicity, not threshold-based, but something like reproductive, developmental, neurotox, systemic, or repeated dose toxicity. Those are the criteria are all threshold-based. So, and we assign high, moderate, low concern for each endpoint. And if the endpoint is in bold color, then that means it's based on the actual analysis of data. If it is in italics, it's based on an analog model or expert judgment. So this is basically an indicator of the level of confidence that we have in the hazard call. And for many chemicals, you'll see there are lots of data gaps and lots of expert judgment is applied in evaluating the chemicals. So this brings it to kind of the question at hand is, if we're handed data from some alternative test system, whether it's ToxCast or some other approach, can we pop it into these these frameworks and just include it in our evaluation. What is the fit like, and what are the challenges that we might have in using these data? So I do want to point out that the rapid test methods, it's really appealing from our point of view in Design for the Environment. Once again, we're making decisions in real time. And so coming to us and saying, well, in 5 years we'll have an answer, isn't helpful to us or to our stakeholders. We have to use whatever information we have in front of us at any given moment to make decisions on a fairly short time scale. So what I'm going to do is I'm just going to put out for discussion a couple different options for beginning to incorporate this information into the types of criteria frameworks that I've been discussing. So the first approach would be more of an evidence-based approach, where you have your results from the evaluation of the high throughput testing or alternative test models. You have some type of pathway altering concentration. Maybe you're able to calculate a dose, but you use this information more as a red flag that says, oh, we have a concern about reproductive toxicity or developmental toxicity or neurotox. We need more testing. And that may be a perfectly valid approach for chemical prioritization, but it's not particularly helpful to someone like me who's faced with dealing with alternatives, assessing and making decisions about alternatives today. So, once again, you have to be very clear about the basis, the context for the analysis that you're doing and the timeline of decisions, because that will make a difference. Now, another option would be to take the high throughput test data and calculate basically a human equivalent dose that is associated with the perturbation that you're seeing. And this is something I know that NCCT is exploring. Can you calculate human equivalent doses based on an assessment of the data from high throughput screening? Now, what we'd still be faced with is the criteria are based on animal dosing, animal exposures. And so there's a little bit of a disconnect there. However, we can convert animal doses to human equivalent doses. This is done every day. It's based on, you're scaling up based on surface area and body weight. And so we could actually convert our criteria to human equivalent and do a direct comparison. Now, that's a lot of different layers of calculations there. Some people may be less comfortable with that because of the potential error that it introduces in the series of multiple calculations. But it's one option. Then the third option I'll put out there is really more of a relative comparative approach, and that is we could take 100 solvents that we've evaluated through Design for the Environment, and we can identify hazard rankings based on our traditional approaches to evaluating chemicals. And then we can take ToxPi scores for each of the endpoints or some other metric that integrates the results of the data, and get a ranking for those same 100 chemicals. Once again, our approach does not involve comparing plasticizers to pesticides, but really it would be comparing solvents to solvents or whatever functional group that we're focused on, and get that relative ranking and asking the question, are the chemicals that we would bin, high concern for reproductive tox, also popping up high concern or the top third of the ToxPi scores, for example, and just doing this kind of bootstrapping approach where you're comparing data to data and seeing if it makes sense. And in that way, we could begin, if someone could deliver us the ToxPi scores, we could begin asking those questions tomorrow and thinking about it, and just does it make sense? If there are differences, well, it could get really interesting there, because who's right, who's wrong, what are we missing, what are they missing, it raises all sorts of hypotheses that can be tested through further research. But at least we could start thinking about this now and how we can start using the data. So in closing, there's no dispute. We need more rapid test methods. We need to move away from the expensive and animal-intensive methods that we're using now. The current criteria clearly are based on animal test data, and so it is a little difficult. It's not intuitive how this is going to fit together, but there are ways that we can begin to start bringing this data to the table and asking some questions about how it can be used, how it can inform our understanding, either through a weight of evidence approach or some type of added value, or at least proof of concept. So I think there's value in doing it, and maybe through this process we will determine that there's an opportunity for a totally new way of thinking about it. Sit down with the chemist and does it make sense, and where does it not make sense, and can we, do we need to do things throw it all out and start again? But before we get to that point, let's begin to do some comparisons and see where we're at. So with that, I'm happy to take any questions.
Thank you, Cal. That was great. So we have time for one question.
It's Bruce Blumberg from UC Irvine. So I like the ideas that you've expressed, and I think that this is a potential way that we can go forward and integrate lots of new data from different approaches into the whole idea of how do we regulate chemicals. The elephant in the room is bisphenol A. Mm-hmm. You saw where it showed up on the tox pie. You know about all the data. You know about the controversy. So I think before you can move this forward, you have to deal with that in some way.
It's really interesting because the methods that we developed really were in this were geared to dealing with chemicals with little data, and so chemicals like bisphenol A present a tremendous challenge for fitting those into these basically hazard screening approaches. Now, full disclosure, I'm in the middle of an alternatives assessment for bisphenol A and 19 alternatives for use in thermal paper. And so pretty soon we will be releasing our draft hazard evaluation, so everyone will have an opportunity to see what this looks like. And yeah, it is I do want to point out that the endocrine endpoint is probably the hardest one for us to evaluate because there's no agreement on a hazard ranking framework for that. So if you see receptor binding, positive receptor binding is at high, moderate, low concern. We're dealing with it more in a narrative approach rather than a hazard ranking approach at this point because we think the data is still too new. It's better to just describe what's out there. So bisphenol, but from a standard endpoint, reproductive, developmental tox, not endocrine, particularly, but apical or frank toxicity endpoints, it is a little bit easier, but you're still dealing with an enormous amount of data, and what do you pick as your basis for evaluation? So it's a challenge. So.
Thank you very much.
Okay, thanks.
Okay. So our next speaker is from across the pond, where there is a somewhat different regulatory environment. We're very pleased to have Sharon Munn with us from the European Commission's Joint Research Center, and in particular, the Institute of Health and Consumer Protection. And Sharon has been, for the last 2 years, coordinating efforts on developing alternatives to animal testing through that institute's ICVAM program. So we're very pleased and happy that you were able to make the trip and tell us about your program. Sharon.
So, thank you very much, Lauren, for the introduction, and
Thank you very much also for inviting me here to speak to you among all these prestigious speakers that we heard yesterday and today. I'm very honoured to be here. And I'd like to tell you a little bit about what we're doing in the EU. To give you an EU perspective, I'm not here to speak on behalf of the whole EU. I'm here to sort of at least tell you what our institute is doing and a little bit about the background, regulatory background over there and some of the activities that are going on. It's that way, isn't it? That's it. Okay. So yes, first I can tell you a little bit about who we are and what we do. The regulatory landscape to set the scene, and something about the emerging technologies and how we want to use them to do more predictive risk assessment, and give you a few actual use cases where we've used some of these new tools in decision making. What is the Joint Research Centre? Well, it's the European Commission's research centre, and we're there really to provide scientific and technical support when it comes to any sort of policy issues and policy decision-making issues. We're there to provide that support. And we're actually, there's about 2,500 people within 7 different institutes over 5 different member countries, but we're actually located in Ispra in Italy, near Lake Maggiore. There's 3 institutes on that site, and our institute is the Institute for Health and Consumer Protection, with about 300 people there. And we provide support in the areas that you can see here. So basically REACH, cosmetics, plant protection products, biocides, food safety, general consumer product safety, supporting different DGs that deal with these different areas actually. But we also deal with a number of cost-cutting issues. You can see here protection of animals used for scientific purposes, safety of nanomaterials our institute is looking into, identification of endocrine disruptors that we've been just talking about, and also this difficult issue of combination effects of chemicals. So we don't work in isolation. You won't be surprised to hear that actually we have a number of collaborations with you guys over here. We're working at the international level with OECD, with WHO, working on the mode of action frameworks from the past, and as well as the OECDQs, our toolbox, etc. But also working in the TOTS21 consortium and contributing to the ILSI-HAZY, RIS21, NextGen, EPA NextGen. So, and indeed, there's our director, Elke Anklam, signing last year with Bob Cavlock, that you might recognise from the picture there, an agreement to facilitate exchange of research results in relation to the ToxCast programme. Well, where are we? This is our current model. This is no surprise to all you toxicologists in the room. We have the animal model, and we use that to predict what might happen in the human. And we spend many discussions on maybe deciding on what the point of departure is for this animal study. But then pretty much we apply a default approach where we're looking at, is this hazardous property? We assume that the hazardous property is relevant to humans, that we're at least as sensitive as the animal, and then we account for variability and uncertainty with our factors. And we've been using that approach for quite some time now. So why change? Well, big driver in the EU is ethical issue, reduced reliance on in vivo tests in animals. But also thinking about it and trying to bring in the green chemistry aspect here, what do the regulators need? Well, we'd be very happy to have faster, cheaper methods to screen many chemicals that are already out in the environment, that they're the bad actors, and we need to risk manage those. But also the product designers, they need the same. I think the same. I mean, you can argue that with me, but the same faster, cheaper methods to screen many products to deselect the bad actors from development and to prevent them getting into the environment in the first place. Of course, this is rather the undesirable scenario that they get out there. What we much prefer is this scenario where it's the product designers that are using these tools up front to prevent those chemicals getting out. In the end, what we're really aiming for is more predictive, accurate risk assessments, and we think we can do that with a better understanding of why the chemicals are toxic in the first place, to move from more observational to more predictive approaches. We have a number of regulatory drivers which give us both challenges and opportunities. The main one, I would say, is this directive on the protection of animals used for scientific purposes.
It's been around since 1986, actually, but last year it was recast and strengthened. And there's a chapter there on avoiding duplication of tests, which obviously reduces use of animals, but also focuses on alternative approaches and says that if there's a test that can give you the same answer but does not involve the use of an animal, then that's what you should use. And it strengthens the whole concept of the replacement, the reduction and refinement of animal tests. And actually ECVAM, which is in my institute, which has been around since 1991 when it was created, it's the European Centre for Validation of Alternative Methods, has now been strengthened in legislation, let's say, because it's been created as an EU reference lab within this directive. But its job is the same. It's to coordinate and promote development and use of alternative methods. This directive itself has then had a knock-on effect, let's say, to bring strong incentives into other product legislation to reduce reliance on animal tests. And the Cosmetics Products Directive has gone a long way to promoting use of alternative approaches by bringing in phasing in a ban on animal testing. And in fact, it's already banned to test cosmetic products and their ingredients in the EU. But for marketing, there was an extension of the ban for the really complex endpoints: repeat dose toxicity, carcinogenicity, toxicokinetics, and including skin sensitization. This 2013's arriving pretty soon. Are we anywhere close to finding replacements in this area? Well, we were asked by DG Sanko to go away and produce a state-of-the-art review. And that's what we did. We invited a lot of experts together in the EU, and they looked into all the currently available alternative methods and came up with a report on the current status. And this was published in Archives of Toxicology this year. The main findings, I should say, is that they don't see there's any possibility of full replacement by 2013 and cannot say when there would be a possibility for full replacement, apart from the area of skin sensitization, which is what we heard yesterday from Ed Carney. One of those methods that you talked about could well be one of the methods in combination with others that actually will find the full replacement for skin sensitization in the coming future. But what they also said was the advanced methods and approaches that are out there now, they hold a lot of promise for the future development of more predictive risk assessment, and this based on improved understanding of how toxic substances reach target cells. But what they stressed was the central importance of toxicokinetics, that this really had been ignored for too long, and if we're really going to be able to predict using risk assessments for decision making, we have to address this extrapolation from the in vitro to the in vivo, and toxicokinetics being the key to that. If we move on to REACH and the incentives provided there, and REACH has been mentioned a lot over the last day, and is innovative in the approach of there were testing annexes, but they weren't called testing annexes. They're called information requirements, and there are standard tests there. But if you can produce the information by another means other than the standard test, then you're invited to do so. And you, in this case, being industry, the companies themselves, the registrants, are invited, if you like, not to do the in vivo animal studies, but to find another way of showing safe use. And really the annex there, the Annex 11, describes what these other methods may be, and they're basically read-across grouping, QSAR, in vitro methods, or a whole weight of evidence approach, so-called integrated testing strategies. And the guidance was produced in 2008 of how to do that. The first deadline for submission of data on the high production volume chemicals was last year. The agency in Helsinki had to, by July of this year, produce a report on what was the outcome and what had been the uptake of, let's say, alternative approaches, which they have done and which you can find on their website there. Basically, indeed, they did find that there was an uptake, particularly of read-across. I mean, it depended on the endpoint, but let's say up to 20 to 30% of the doses that they got had used read-across arguments. rather less was use of QSARs. Let's say 1 or 2% had used QSARs. And also there's the possibility for waiving if you're saying that the substance will not get into the body. So this is the bioavailability issue brought up by Adelina yesterday, that really if you can demonstrate it's not going to get in the body, then you don't need to do the assay. So they also said that, of course, at the moment they have not gone in, that in the dossier compliance they're looking at whether you have to justify your use of read-across. To the extent to which this read-across is going to be accepted, there's a suggestion in the report that it probably is not so well justified in some cases, and that they will in some cases probably be going back and asking for further justifications, or even in the end asking for the test. Other drivers that are really hot issues at the moment, both sides of the pond, endocrine disruptors, it's clear. We have some deadlines in our legislation coming up. 2013 is the magic year. By the end of 2013, we must present criteria for evaluation of substances to identify possible endocrine disrupting properties. That's for the Pesticides Directive. Similarly, in REACH, end of 2013, we need to come up and decide whether this issue of threshold or non-threshold, are we really going to say that we can adequately control substances that have been identified as endocrine disruptors, or are we going to say that we can't go down the adequate control route, we must look into the socio-economic benefits of a substance and look for substitution of that substance? So that's the discussion going on there for REACH. And the biocides regulation, that's also being recast at the moment, and they're pulling in those issues as well in a similar way. And mixtures is also high on our agenda. Back in 2009, the Council came to the Commission and said, look, we have to do a better job here on mixtures. You can't keep ignoring that we're all exposed to mixtures and not single chemicals. So a state-of-the-art report was made, and now our scientific committee on emerging and newly identified health risks is actually looking into that state-of-the-art report and going to produce an opinion on their views on it very soon. But basically, beginning of 2012, the Commission has to reply to the Council, and the Danish presidency is next year, and they put it on the table in the first place, so they're really going to be driving this issue at the beginning of 2012 on mixtures. So back down to something a bit more basic and practical, maybe, in terms of the screening and prioritisation and where we might have some use cases here. There are situations where we cannot ask for testing, or it's just impractical. And in this case, we need these predictive tools for that reason. And we can see there that there are some specific criteria that we have to avoid. And Carl brought it up earlier, that we know that we have to avoid persistency, bioaccumulation, genotoxicity, and we can more or less identify those without doing a huge amount of effort. We can have an idea of which chemicals are going to be persistent. We can look at the log KOW, etc., and have some good ideas pretty soon of what we should pull out and what we should avoid. We have used some computational approaches in these areas that I've listed here, and I'll come back to those later. In the area of identifying chemicals to be identified as priority hazardous substances under the Water Framework Directive, in assessment of pesticide metabolites in feed, where we can't get the data on the metabolite, and also in the future when we get to the lower tonnage substances for evaluation under REACH, we may well find that some screening tools are very useful for that purpose. So what's new? What emerging technologies have we got there, and how can we apply them to more predictive risk assessment? This covers really a lot of the ground that some of the speakers covered yesterday, I would say. We have all these tools available to us, and how can we use them is what we're looking at. I mean, over in the right-hand corner, you have the whole genomics aspect of profiling for protein expression, and the normal in the cell, the normal sort of structure of the cell. This is what it's doing all the time, of course, and we have to understand what is a perturbation, I think, there, and that's where we're struggling in that area. But we're also working on the human stem cells. We've got the possibility now with these induced pluripotent stem cells, and we can differentiate them into all different types of cells, and that's really exciting, that we might be able to really do some good in vitro work on cells that have really come from a human origin. Of course, these cells like to be in a 3D environment. That's what they're used to in the body, and we now have possibilities to start building these scaffolds that we can really have 3D structures. Automation of in vitro methods, we have the possibility to screen for many hundreds, if not thousands, of chemicals there, and not just looking at high throughput, but also the high content now. Under microscope, we can look at many things going on in the cell at the same time. And all these data can feed into our computational models. I've pulled out PBPK models again at the top there because this is, again, very important. Maybe not so much from the screening hazard ID aspect, but certainly if we want to use them in risk assessment in the end, we need these models. And to a certain extent, we're dabbling or delving to a greater or less extent in each of these areas within our institute. So I'll just tell you briefly a little bit about a couple of things we're doing there. So we want to, maybe no surprise here, to embrace the whole strategy of a mode of action-based approach. So really looking at what is the mechanisms behind what these chemicals are actually doing in the body. And then we can design our systems, in vitro systems, around that. So if we can take the in vivo data that we've got from wherever we can get it, we can, from that point of view, look into and try and identify modes of action. And then we can really see what are those critical events, and then design in vitro tests around those critical events. And then maybe from there, if we can make the case for where we have the reference chemicals and we can make the connection from the in vitro in vivo, then we can look at the structures, and then we can maybe do some read across to some other chemicals, and we can look at those same assays for those chemicals and see if the whole thing hangs together, and we can see the correlations are there. Important thing really is that we want to use the in vitro, the in silico, the in vivo in combination, and have the learnings from each and keep feeding that back into our models as we're developing them. So I was glad to see Adelina use this slide yesterday just to remind ourselves this is not easy. These are all the biochemical pathways that we know about in the human body. We've used rat as our model to encompass all of these in the past in our black box. So we have a disturbance here, and we have a toxicological effect somewhere else. And in between, we have a highly complex interaction of many pathways, and that's what we have to try and grapple with. So maybe we have to sort of stand back a little bit and say, okay, let's take one thing at a time. Let's look maybe at liver toxicity. Chemicals get into the body. They're very often, they're toxic to the liver. That's where they hit first, but certainly by the oral route. What do we know about liver toxicity, and looking at all the different mechanisms for toxicity? And this is the way we're trying to work now in terms of understanding which critical events or intermediate events we want to recapitulate in vitro. So we have in our laboratory now, we have some very nice human hepatocytes that are metabolically competent, so we want to use those. We have the possibility to do automated cell imaging where we can look at many different types of endpoints. And so this is where we're just starting off some work in the laboratory now with these cells. And this is what we do. We culture and differentiate. Then we treat the cells, and we run them through our high-throughput screening here. High throughput in our system is 92 chemicals here. We're not doing thousands at one time, but we can measure, let's say, high-content imaging. We can measure a lot of different parameters at one time and analyze the data. And then we want to categorize them according to sort of profile the chemicals according to which events they're sort of turning on in the cells. Just to say that we have some other assays that we're looking into, the microelectrode arrays. Here we're measuring electrical activity in neuronal cells, so we're not just looking at straightforward cytotoxicity. We're really able to culture these rat embryonic cortical neurons. We can measure sub-cytotoxic concentrations. And so it's a real functional assay. These cells are living, they're connecting, communicating, and can actually monitor effects over time. So in trying to look at more than just acute toxicity, we can look at, try to recapitulate more low-dose chronic exposure. And we also have the possibility here to look at mixture effects in these cells. This is a more sort of higher level, more complex assay. It's still in vitro, but it would not at the moment be amenable to automation, so we couldn't do many chemicals this way. But they can be useful for certain circumstances when we suspect neurotoxicity. And this just describes how we do it. We have this chip which has the electrodes in, little plastic chip with electrodes, and then we culture the cells for 21 days in there, and then you can really, they grow together and they communicate together, and then you can really measure their activity and analyze the data from the signal, and from that you can work out the firing rate and the effect of a chemical in reducing or inhibiting that firing rate. So this was just to mention the whole area of computational toxicology that we're dealing with, the read across, as well as the mathematical models and the PBPK models. That was really to bring the point that we should be exploiting all the existing data as far as we can. And what's important is the data management and data analysis, and making tools available that you can go in and interrogate these databases. And I just wanted to give you one example of some other work that's been going on in our labs, which is a relatively simple approach which could be really useful in sort of screening chemicals. Because here you've got a sort of a PBPK model that's really focusing on human bioconcentration factor, so that you can really, here you can see that you can rank chemicals according to their likelihood to bioconcentrate in humans, and obviously the red part is maybe the chemicals you might want to avoid because they were the ones that are more likely to bioconcentrate. So panning out a bit from what we've been doing in our own laboratories, more to excuse me, more to the whole of the activities in the EU in relation to funding of research into finding alternative approaches. We have the Accelerate activity, and this coordinates all the research that's been doing that is being done in Europe that may be contributing to finding replacement to animals, and it's really working along a toxicity pathway approach and trying to bring that in, and it brings all these projects together. The coordinators of the project come together, they exchange ideas and results, and they hopefully can then move forward better when they're really speaking to each other, because we found in the past that we've had a lot of projects going on in the EU, but they're not really talking to each other, and this way we hope we really can make more progress if we get people speaking to each other more. And also in relation to what I mentioned before on the cosmetics, and this marketing ban coming in 2013, the cosmetics industry, Colipo, are very interested to form a public-private partnership with the Commission to both put in 25 million euro to fund some projects to really try to move forward in this area of repeated dose toxicity. So you see safety assessment ultimately replacing animal testing. So we've got 6 projects, 70 institutions involved. The project started in January 2011, runs over 5 years. And we have one coordination action that would do a little bit like the Accelerate project. It's to make sure that everyone's working together and exchanging information. And again, with this strategy, really up front is this toxicological mode of action framework that we're trying to put over the whole, imbue in everybody from the starting point. When you're looking at your reference chemicals for your testing, you're really thinking, getting information on those. How are they creating their toxicity? So that you can really build it from a mechanism basis when you're designing your assays. These are the projects. You can see they range from stem cells through to human biomarkers, computational tools, organotypic human cell cultures, And interestingly, we also have a project there which is really there to capture the data, analyse the data, and make the data available, the ToxBank, and the coach is the coordinating project. So I dare to put up the word validation. I know yesterday it was we've changed it to verification, apparently. That's okay, we can call it verification as well. But just to point out, of course, it's validation, verification of your results is an intrinsic part of any scientific process. And really it should be fit for purpose depending on what you're going to be using the outcome for. If you're going to be using it in regulatory decision making, obviously you need to have the regulators will need a lot of confidence in that result if it's going to be the one data point that they're going to use to make a decision on. But if you're going to use it for screening or weight of evidence approach, maybe the extent of validation does not need to be so great. We have the ECVAM modular approach here, which can be picked up by anybody and basically used as seen fit. But these are the basic concepts of what you should do when you're validating your test. Obviously, in Europe, we are really designing assays that can be used by not just by one laboratory, but by many laboratories. So in that sense, we need to be able to transfer it to other laboratories and show that it works. But most importance comes down here at the relevance side. And of course, with a lot of these new assays, we're having the big discussion of what are we validating against? What are we predicting for? What is our reference that we're predicting for? And that can is proving quite interesting discussion. Of course, in terms of, again, regulatory decision making, some of these assays may need to go through real formal validation, independent peer review before they would be accepted as an OECD test guideline, etc. But actually, in our laboratories, we've been involved using the high-throughput screening not just for developing assays, not just for generating data, let's say, but they're also been used in the validation context. And we've been involved, for example, for the NRU 323 cytotoxicity assay, they're in upscaling it to an automation platform and involved in the formal validation study for that assay. And again, also for the Lumicell. This is a reporter gene assay for estrogen receptor binding, estrogen receptor transactivation. The validation study is run by ICVAM NICETM. It's now finalised, and it's going forwards for an OECD test guideline. But we have also been involved in upscaling and automating it to our platform in our laboratory. There's just a couple of other things before I close that I'd like to cover. One is this at the OECD, with a view to the future and using more of these upstream in vitro tests in our decision making. We felt we needed to find a way of capturing that data. And at the moment, of course, most of the harmonized templates are designed for your standard toxicology studies related to an apical endpoint. But we felt we needed something, if we're going to be more upstream, and the fact that an event could be related to multiple apical endpoints, we really needed to come up with a template where we could record intermediate effects and then link them to different apical endpoints. So that's what we're aiming to do with this harmonized template here. First of all, we want to build this taxonomy of possible key events or intermediate effects that we're calling them, which might ultimately be identified as key events, because we don't know at this point in time, but we want the taxonomy there, and then the possibility to link to all types of different observed effects or apical endpoints. And we're doing this now. We will be having a meeting quite soon in the Joint Research Centre where we've invited external experts, including well-known people from US EPA, to come over and give us their view on this, and then we want to present it to the OECD at the beginning of next year. Coming back to some more use cases, as I said, in hazard assessment, we have various, mainly it's been the computational area, I would say. The SARS and the QSARs of what have been actually used in decision making so far, for where we've not been able to generate in vivo. And one of them, just to show you one example here, where you can do quite let's say, simple exercise using these sorts of models to predict persistency, bioaccumulation, and toxicity. On the other side, you have some other similar tools to predict exposure, and that can give you a handle then on a ranking for the potential toxicity of these chemicals. And we did it on over 2,000 substances, and from that, the Water Framework Directive basically identified 78 of those as potentially hazardous substances from the 2,000, using this approach to fill the data gaps. And this is a slide. I just wanted to put this in here because this is European Partnership for Alternative Approaches to Animal Testing. This is where the Commission again has partnered with industry across 7 industrial sectors, so from pharma to pesticides, industrial chemicals, cosmetics, really to work together on finding solutions. And I think this is very important because obviously we know, for example, that the pharmaceutical industry really have been using a lot of these assays in their screening, and we want to sort of find some ways of sharing that data and that knowledge in a way that doesn't involve them exposing confidential information, let's say, but to really find best practice by building on everybody's knowledge in the area, and that's really what it's trying to do. So just to conclude, we have in Europe strong regulatory drivers to find alternative approaches that are less reliant on animals. We want to adopt this toxicological mode of action framework as a strategy in which to apply our new knowledge and tools that are out there. We have to recognise the importance of the reliability and relevance of results in the context in which you're using that data. And we want to bring all these tools to bear in an integrated manner. We want to increase our interaction between the disciplines and also this industry cross-sector sharing of ideas and data. And last but not least, at least maybe not in the hazard ID screening aspect, but certainly when we're coming to use these in the end for risk assessment, which is what we're trying to do, then we have to be able to do this in vitro to in vivo extrapolation, which depends on this aspect of the ADME and toxicokinetics. So with that, I'd like to thank you all for your attention, and this is where this is a joint research centre in Italy by Lake Maggiore, so you're welcome to come and visit us there anyway, any time you like. And if you have any questions, I'd be happy to answer them.
Thank you.
I'm sure many of us would like to come and join you there. I think we have time for one quick question. I see Richard at the microphone. Richard?
Thanks, Richard Dennison, Environmental Defense Fund. Sharon, I wanted to test whether I'm detecting a somewhat different philosophy or approach in Europe relative to the U.S., and I'm thinking in particular of ToxCast, maybe, and if David's in the room. It seemed to me like you were saying there's more of a targeted, focused approach. You're not necessarily using every assay you can find on every chemical you can get. And in the U.S., perhaps, without meaning to sound pejorative, a little more of a brute force approach where you just generate as much data as you can, as many assays on as many chemicals. Is that a real difference that you detect between the approaches, and could you say a bit more about it if it is?
Well, I think it's not bad to try many different approaches. And we're working with, we've had David's been over to our laboratories for discussions on what they're doing, and we're telling them what we're doing. And we're trying to work together because we want to add something additional to what you guys over here are doing. And we appreciate that it's great, and you have the possibility to screen all these thousands of chemicals in these assays, and that you're going to be able to see something interesting coming out of that. But there's different ways as well of approaching this, and we are focusing more on coming from the, let's say, focusing upstream and then moving backwards and seeing if we can come to the point of selecting certain key events more upstream from the omics type data, because the omics type data, we're not sure at the moment what that there's a big space in between, between the perturbations at that level and the actual adverse outcome. So we're trying to look in between at the moment, and that I think will be complementary in the end, or it's meant to be. So, David, I don't know if you want to comment back.
I agree.
So our next speaker is Tom Osmuth. He comes with 25 years of experience in safety assessment and product development. He's the president and founder of Science Strategies, a consulting firm. Previously, he was vice president of global safety assessment and regulatory affairs and sustainable product innovation for SC Johnson. He co-chaired EPA's endocrine disruptor methods validation advisory committee, and he served on EdStack, so a lot of experience at different levels in dealing with this kind of big issue. So welcome, Tom.
Thanks, Lauren. I appreciate it very much. And thanks for the opportunity to talk. Again, I come at this from a very different position, I think, than most people in this room, and I'm pretty humbled to be around the people that are really digging in deep on this issue and doing tremendous things, I think, in the science. I have this right? Let's see. There it is. This way. Okay. What I want to do is talk a little bit about some of the challenges. In the agenda, it says bridging, and I don't want to use that word too much, but there are a lot of gaps that we need to fill, a lot of things that I see from my perspective that would help advance this field and help advance the application of it. And in our consulting practice, we deal with very large companies, some of whom are in this room, down to very small companies that have new molecules, or they've had private funding, don't even have a public offering yet, but they're all trying to grapple with the issue of producing safer chemicals, defined many different ways. So we're actually on the ground trying to help people apply these techniques now and try to help people realize there's a lot of uncertainty, but still make intelligent decisions about product development. And that includes not just producing new molecules, which we do some work with as well, but also choosing among the many molecules that are out there. So in addition to dealing with questions of developing molecules, how do you help companies decide between a myriad of choices, many of whom those companies have very little experience in doing that and often very little technical capability. We're working extensively with the toy industry globally to deal with the Toy Safety Directive in Europe. That's where a lot of our work is right now. And they are an industry that has an awful lot of concern about safety, and clearly they present the opportunity for exposure to children. But from a technical and scientific standpoint, they don't have the sophistication of most of the people in this room. So my perspective is one of dealing with a lot of different stakeholders and trying to be practical and being immediate about being able to do some things. There's some scientific challenges we'll talk about. The idea of data versus understanding and wisdom, talking about bridging the old to new toxicology, identification of test batteries. We heard a little bit about this before, but I'll give a couple different perspectives, and also talk about some of the cultural challenges that I've seen in dealing with this issue. The other thing I really love about what we do is we deal with people with all kinds of backgrounds, deal with business people, marketing people, investment people, scientists, toxicologists, all different kinds of folks, so we get different perspectives, and that's where the cultural aspects come in. Scientific challenges. I love this quote. It applies to so many things. Data are not information. Information is not knowledge. Knowledge is not understanding. And understanding is not wisdom. I hear from many people that there's lots of data, there's lots of information available. There is, but there's a lot more data than there is information, and there's a big difference. It's being produced at low cost. There's some work that we're doing where we're screening a number of molecules for potential modes of action relating to carcinogenesis, and we can get so much data in such a short amount of time. It is just remarkable. I still don't exactly know what to do with it yet, and even some of the statisticians we work in have a hard time explaining the application of it, but it's pretty exciting. There's an interesting blog by Amy Palmer on a site called ingenuity.com. She talks about Zen and the art of complex biology. And the whole purpose of Zen is trying to get rid of noise and reduce everything to the essential elements. And that really describes a little bit of what we're trying to do with regard to the techniques that have been presented in this workshop. And certainly when it comes to looking at some of the data, that's what we have to do. I'm just listing some of the various assays that are available that NIH is working on. You've heard about some of this before, but maybe not all of them. The high throughput screens, there's some interesting ones here that also are available commercially. You can see that many of these can be done very quickly. You get lots of information, lots of data, I should say, and if you put it together properly, you can get some information, maybe even some knowledge. I think you're going to see these being applied more and more, not just by the chemical producers, but by also chemical choosers, and that would be companies that can select between different molecules, because these are relatively straightforward, relatively inexpensive compared to whole animal studies, and they provide an awful lot of grist, at least for thinking about it. This is the kind of the classic ToxCast slides, and you can see what we're doing in toxicology is moving from this area where we used to be doing all whole animal work back down to thinking about some of the basic properties of molecules, the molecular descriptors that Adelina talked about, and then to some of the more applied things. And I think this is very exciting, but the challenge it presents scientifically is not insignificant. And I guess this is an example of some data. We're doing a lot of work trying to understand rodent carcinogenesis that relates to humans. And how does that fit into green chemistry? Well, there's only so much resources around. And if we spend a lot of time and resources chasing effects or chasing results which have been indicated by animal studies that aren't relevant to humans, those are resources that can't go into developing safer chemicals. So a lot of the techniques that I've had personal experience with is trying to understand what's relevant in animal studies and what isn't. And what that really gets at is the basic pathways and how those pathways are different between animals and humans. The better you understand the humans, then you can use those screens for predicting what would happen in humans. But this is just an example of all the different gene symbols, all the different genes you get data on from a fairly high throughput assay. And the numbers over here are just fold changes in response to the various treatments. And this is dealing primarily with CAR and PXR, as it relates to non-genotoxic carcinogens in liver. But information is what we care about even more than just those data. How do these changes relate to specific pathways? These are the ultimate eye charts, and I think Adelina and Sharon, I can outdo you because for almost every one of those steps along the way, different metabolic effects have a whole cascade of gene expression impacts. This is just one fairly simple one about DNA damage. This is the one that we deal a lot with, chemical oxidative stress. And one of the ways of expressing the data, is there a pointer on here? Lauren, is there pointers at the... Oh, yeah. Right on here. Aha. Let's see here. Very top. And let's try this one. So it's on. Here we go. And the way these... I kind of like the way that they express these because what we have are little thermometers. And red means upregulated, blue means downregulated. But you can get from these assays, the ones I showed you earlier, information on a lot of these expressions. And not being a systems biologist, though I'm trying to learn as much as I can in a hurry, you still get some pretty good indications of when a chemical comes in and what it affects, not just in isolation, but how it relates to the whole pathway. That's a big improvement. Here's another complex pathway that I won't talk about. Now, if we want to get what I would call knowledge, and this is trying to understand the relative effects of these various pathways operating at the same time, and then start to make some relevance out of it for a whole animal, here's one way of expressing the data as well. And this is, ignore the actual chemicals here because they aren't that important, but what you end up seeing is if we take a look at all those initial genes I showed you can be grouped into whether they're related to CAR or PXR, and not on that chart, but another data chart, we have PPAR alpha. And you can take a look at the expression following exposure to phenobarbital or a test article, various days, and what you see, for example, here. And this is actually a pretty nice way to show it. High induction here, and what you're looking for is similarities between expression with the test article versus the reference compound phenobarbital. And you can see here, especially out at 30 days, you actually get repression of the cholesterol biosynthesis genes with the test article versus phenobarbital. You see similarities in their effect on CAR, and you see, some differences here in oxidative stress, and then a fairly high induction of the genes associated with signaling and DNA damage. This is starting to become something that biologists and toxicologists can relate to and start thinking about when it comes to whole animals. This is part of the goal. This is close to the goal of what we want to do. What we really want is, understanding. And that is really where you integrate this and have a relationship with these data to humans especially, or to wildlife ultimately, all living organisms. And that is tremendous challenge. wisdom is what we want to get out of all of this, and that is the ability to make smart choices about using chemicals. That's why we're all here. That's what Paul and Assays talked about. That's the whole genesis and the purpose for green chemistry. It's easy for me to get lost into the pages and pages of gene expression, fold increase charts that we see, and lost in that. But this is what we ultimately want to do. There's a lot of uncertainty, because just think of all the steps along the way. There's uncertainty in all those data. I think Cal, I agree with her entirely. You want to have some directional indications which way is better than where we are now. And we have to realize this is going to be a very iterative process with a lot of continuous improvement guiding it, as opposed to absolute bright lines and yes, no. So one nice example, and let me back up here. This is the same table I think that Cal showed, actually. And I like this table because it really is, and this is a relatively early, but it's a good attempt of trying to integrate a lot of different things and still somewhat parsimonious. I think Helen mentioned an excellent point yesterday. We were talking about at dinner last night with some other folks about, I would just call it parsimony of decision-making paradigms. It's so easy to get wrapped up in, and I've done this myself, especially when you get quantitative, in really complex schemes and paradigms. I'll show you a couple a little bit later that can do that. But at the end of the day, you've got to make a decision, and it's going to be a go, no-go decision. This is better than that. This is not as preferable, etc. And I think this is a pretty good attempt at integrating, and Cal went through the details of that. But this is when you start getting to make wise choices, when you can start doing this with the data. Okay, other scientific challenges that we have to deal with, defining what is green. I don't even want to go into detail on that, because that's a difficult value-laden discussion, which is part science, part values, and to me is not necessarily too illuminating at the moment. I certainly agree that we have to have some kind of a battery, a combination of things. I always caution about ascribing too much precision to numbers, and that goes from everything from animal toxicology studies to gene expression, fold increases. It's really scary to think of seeing numbers expressed to 2 and 3 decimal points in systems that just are not that precise. And let me make a comment about traditional toxicology, because that's where we're starting, and that's often our reference point. We tend to use uncertainty factors, or assessment factors, as they call them in the EU. And I think I'm very encouraged about where we are now. We still have to bridge back to that. We have to use that as kind of our grounding. But if you think of where a lot of toxicology is, and I'm not talking about the people in this room, these are the leaders in this room, I will say. Much of toxicology still involves doing a very expensive study, getting a no-effect level, and then dividing by 10 or 100 or 1,000. And my 14-year-old, he's now 15, but last year he's asking me what I was doing, and I was talking about toxicology studies, and I said you do this dose response curve, and you take this point here, and then you divide, and you get this number, and it's safe. And he said to me, well, what about all these animals up here? Why did you kill those animals up here on the other part of the dose response curve? And I started thinking, yeah, you throw out all of the data when you do this no effect level divided by 10. And benchmark dose is certainly an improvement, and that's been around for a long time, but still, it's hard to find some people in regulated communities who understand benchmark doses and understand their applicability. But his question was a good one, because what I think we need to do is bridge the very simple approach that a lot of regulatory folks are taking, and again, the people in this room are exceptional because I don't think they approach it that way, and move to something much more complex. That is a huge bridge. Some of the difficulty that we've had in just talking about benchmark doses as opposed to the NOEL divided by 10 and 100 or 1,000, those can be difficult discussions, but moving beyond that is something we really have to work hard to do. And unless we make that bridge, we won't see the application of these data the way they need to be applied. Oops. Okay. Old to new, I mentioned that is a huge challenge. It's a challenge not just for people in this room, because I think here people are making that transition, but the Society of Toxicology, a lot of toxicologists who are trained in the traditional toxicology world, which really isn't that long ago, and still much of it still is, being able to be comfortable with these numbers, being able to be comfortable with the tremendous amount of data, and systems biology is something that we need to do. Regulatory applications, again, I think that's going to come, and that always lags science, but it needs to be pushed very, very hard. And then finally, I'll talk a little bit more a little later about the idea of incorporating the hazard findings into a broader context. Again, that's the ultimate wisdom piece. Okay, cultural challenges. And these are very important because I see this all the time. And again, what I love about our interaction is I often am in a room where there's maybe 1 or 2 toxicologists, then 15 other folks. There is a precedent. And I go back to, in fact, I was sitting at College of William and Mary, and my middle son's girlfriend was starring in Oklahoma. And they're singing this song, and it was not long after the SOT meeting. And I was thinking about not farmers and cowmen, but I was thinking about chemists and toxicologists. And it's interesting because we do two different kinds of things. Toxicologists like to study harmful effects. Chemists like to synthesize new molecules. And they're very smart at both of those. But really, we do have to stick together, because only through sticking together can we advance the whole field of green chemistry. I think it's great that toxicologists have been invited into the field, and I certainly see a lot of folks here and elsewhere that are excited about that. But it's absolutely essential that they work together. So from a toxicology standpoint, I think befriending, or I guess as people under the age of 20 would say, friend a smart scientist. I don't think of friend as being a verb, but that's okay. A lot of nouns have been verbed recently, and that's just another one. Befriend a smart chemist, becoming familiar with the existing design rules, and Adelina presented some of that yesterday. It's amazing how many folks in toxicology don't think that way. Again, a lot of folks, are pretty much stuck in we do this study, we get the no effect level, we divide by 100, we pass on, we do the next study, et cetera, et cetera, et cetera. Because that's really what the ultimate regulatory requirements are, especially in the regulated areas such as pesticides, where a lot of data development occurs. Maximizing the value of every study that's run is really important, again, for helping bridge this gap between the old and new toxicology. In vitro assays, wonderful. They're relatively inexpensive, and when I work with companies that have maybe 12 analogs, and thinking of one example, actually a bio-based company that has maybe a dozen analogs, probably 5 or 6 of them are commercially relevant, but I encourage them to run all 12 because the knowledge you gain from the structure activity standpoint is valuable, and it's relatively inexpensive. They chose to run 9, but that's okay. It still is better than just doing 6. So if you have a chance to run more inexpensive high throughput assays, do that so we get more information, more data to use. In vivo studies, again, I'm preaching to the choir here, but never, ever, ever, almost never, run repeated dose studies without considering internal dose. Again, it's just, to me, it's a shame to waste all these animals and do nothing more but do clinical chemistry, body weights, organ rates, and histopath. There is so much information that we're wasting. And the most important information, and we've heard allusion to it in other talks, is the internal dose. Because that, again, is really, that's the concentration that people are getting exposed to. It's the target tissue, we need to know that and need to understand that. So this is really essential. And for any kind of future model development with PBPK, in vitro to in vivo extrapolation, we need to have this. And especially because we have to get beyond just the NOEL benchmark dose. I think we are. All the people in this room, I think, understand that. But when you get to the broader regulated community, I don't think it's there yet. Okay, chemists. Befriend a smart toxicologist. Becoming familiar with design rules. I think some chemists are, certainly the ones that are in the green chemistry world, but that's a small portion of chemists. Take a short course in toxicology. People have mentioned that before, but I've talked to some chemists at various companies, and their toxicology knowledge is limited to an MSDS. That's really about it, because that's really all they had to learn. And when you're in school, you do what you have to do, and you don't have a whole lot of time for much else. So that's something that is changing, but it needs to change a lot quicker. The other thing I think that has to happen, and it doesn't have to happen on an international EU, US, global interaction standpoint, is just at a very low level collaborate with the toxicologist and talk about chemical analogs you can synthesize. And they can be useful, relatively inexpensive. They can be very useful to do this. And maybe you can try to see if you'd predict there'd be a reduced hazard. Look at those design rules and try some things out. It's hard sometimes in the constraints of a large company to do this, but I think the future of advancing here depends not just on the large, somewhat cumbersome regulatory research establishment. It moves very well. It kind of moves slowly. I think it moves effectively, but there's a lot that can be jump-started by just 2 people getting together and trying something. I think we're going to see more and more of that, and if you try something that's interesting, get it published. I tend to be a believer not just in the big research institutions and large corporations, but there's tremendous innovation and creativity in small organizations and ad hoc relationships that I think really is one of the most dynamic aspects of the American economy, and it's something that really needs to be encouraged. And in this area, we can make huge strides. All right, risk versus hazard. This is another cultural issue that we deal with all the time, or I certainly run into. Risk matters and hazard matters. They don't all matter equally and don't all matter the same at the same situation. It's easy. I've run into folks in large corporations when they talk about green chemistry or DFE. They think, oh, it's all hazard-based. I don't want to deal with that because it's just, it's unreasonable. Again, the people in this room, I don't think feel that way. This is a group of people that understand gray areas very well. But if that kind of viewpoint really, and I call it being the perfect, being the enemy of the good, you hinder continuous improvement, and we have to acknowledge that there's a role for each one of these. I'll talk a little bit more about that. Actually, I'll give an example I think coming up, is you can use risk to prioritize, and that can be important, especially when you start thinking about balancing the hazard criteria with other criteria for greenness or sustainability. And you can consider both the hazard and the potency, and potency is important, and that's really the dose response. Something that's not very potent, you probably don't care about as much as something that is. So if you have limited resources and attention, I would put it on something that is more potent. Making the business case is always difficult. The companies in here, I think, get it for the most part, but it's never easy because it doesn't always work. The numbers don't always pay off because you can't always estimate intangibles and you can't predict what would happen if you didn't do something. And that's just a challenge. There's also an uncertain competitive advantage. Companies have said to me, well, why would we do this? Because someone else is doing that. And by the way, our upstream and our integrated manufacturing supports this kind of chemistry, so how could we change and why would we change? Those are challenges that are not easy to address, but for the companies that have done this, it really has taken a commitment from the very top of the corporation because it gives everybody underneath the top permission and license to operate. And that was my situation at SC Johnson, where the chairman at the time, Sam Johnson, was on the board of the Nature Conservancy, was fairly environmental oriented, very community oriented, and we always knew that we had his cover because if it was something that advanced sustainability, advanced product development in a greener way, that was something that you had permission to do. So that's a very important cultural thing for this to work. Okay, integrating this. Do we need to do that? Well, yeah, we do, because there's more to think about than just hazard. Although as a toxicologist, I think that's generally the most important thing to think about, but it's not always true. You do have to consider efficacy and economy, meaning just cost, resources, et cetera. Is it something that's feasible? And, again, I said hazard is important, but it's not always paramount. And here's an example. I think this is actually from Richard Judson, who I think is in the audience, presentation at SOT, where actually using some of the new techniques, we're able to combine some, if you will, dose response or activating concentration levels with predicted internal dose. And what you can do with that is produce something not unlike this. And the point here, and this is actually from this paper about a year ago, is a way of prioritizing what chemicals are of greatest concern. Now, these are all pesticides because there's a heck of a lot of data on it. And this is related, I believe, to endocrine-related activations. But what you have here is a mean, I think, geometric log scale, yeah. So geometric standard deviation about these various activities that were measured from some of the high throughput assays. You have this value here converted to a milligram per kilogram per day dose, estimated in animals, and then you have down here estimates of actual human exposure. So what this would say is if you look at this material here and this one that many are familiar with, triclosan, the exposure here is within a couple standard deviations at least of where you might see some significant effect, or at least an AC50, I think it is, of the actual material in one of these assays. This isn't perfect, and you can't necessarily use this to regulate, but you can use it for some kind of priority setting. And, oops, there we go. Now, when you integrate this, the ideal result is something like this. And Bill Greggs gave this presentation last week. Bill used to be with Procter & Gamble and now is working with grocery manufacturers. Gave this at the California meeting on alternatives assessment last week. This is a relatively simple-looking scheme. but it gets pretty complicated when you start thinking about what numbers you put in here for economic impact, consumer assessment. How you rate these is very important. But this is an attempt, again, to integrate everything, and we need to try to come up with a framework to do that. This is the ideal result. Here's your baseline. It's not so good because it has some effect on sensitive subpopulations. Let's say children. Alternative one, you don't have those worries. You got a little bit here, maybe on functional performance, maybe some issues here. That's not so good. Alternative 4 looks great. Alternative 5, downscale on energy efficiency. This is the perfect world, but that is probably not going to happen. This is what you'll often end up with. And balancing these, there's reds everywhere. You can't get away from it. There's always trade-offs. Balancing these and making intelligent decisions, this is something that DFE has certainly struggled with regard to their alternatives analysis. I think they've done a very good job and they're very transparent about how they've done it. Unfortunately, it's hard to scale DFE. I mean, you do the best you can with what you have, and they're very challenging assessments. But this runs into some of the issues we have to deal with, and maybe in the panel discussion we'll address some of that. Okay, final perspective. George Box, statistician, University of Wisconsin, said about models, all models are wrong, but some are useful, and the same applies to all of these scoring schemes. There's nothing perfect, but they're all useful and they all have their value, as long as the data underneath them are good. And that's, again, where I always come back to, I can probably work with any scheme, but I need to understand the data and have it be valuable. So in closing, I do think we're in the golden age of toxicology. It is really exciting to be involved in this field. And I feel fortunate to be in a room with people who really are, it's easy to forget that, but you're truly at the leading edge of this field. And you never know where it's leading sometimes, which is exciting. But the challenge, again, to use that first quote, is to use these data to develop information and knowledge so you can understand the likelihood of adverse effects in whole animals and the environment so you can make wise decisions. And that's really what green chemistry is all about. So with that, I just want to acknowledge some people who are very influential to my interest in the field. Paul Anastas, Adelina Fachkova, and Julie Zimmerman, who befriended a toxicologist, that being me, at Yale, and helped me get involved in the whole issue of dealing with the design of safer chemicals. Bob Olfing and Steve DeVita, who I don't work with personally, but they were some of the first people and still continue to advance that field, and they published a book, almost 20 years now ago, on designing safer chemicals. So with that, some more resources, and thank you for your attention.
Thank you very much. We have time for one question. Okay. You raised a number of provocative issues, but we'll be taking it up in the panel discussion. Thank you very much.
Thank you.
Okay, so we'll take a break now. If we can be back here in 10 minutes at about 10:20.